Hello everyone. I am Elias Zuchelli, a master's student with University of Tennessee under the Plant Pathology program. And today we'll be talking about how we can keep the fungicide resistance in check. To have a better explanation about the fungicide resistance, we need to go over the fungicide history. And the first fungicides that have been used in the field were way before we even were born, or when some of you still had hair on your head. But the majority of those fungicides, they were multi-site fungicides. So they have a broad spectrum of action in your fungi, what means that they will act in several activities that are lethal to the fungi, and they will need a higher rate to be efficient, and also they will be more toxic to the environment as well, and they will not be as efficient as the fungicides that we have nowadays. But on early 90s, we started to have new products, single-site products though. They had just one point of action in our fungi, but they are much more efficient. We need a lower rate to them control our pathogens and our disease in the field. Therefore, four classes of those fungicides are mostly used for row crops, and between them we can point out KOI fungicides, which act in the fungi respiration, and they are most effective as a protective fungicide, which will inhibit the spore germination. Also, these fungicides are classified as a high-risk disease development by FRAC. Besides the KOI, we have the SDHI fungicides, which also act in the process of the respiration of the fungi, a slightly different point of that process, which make those fungicides medium risk to develop resistance. And also, besides the KOI and the SDHIs, we have DMIs. DMIs work more after the fungi has infected the leaf tissue, breaking down the fungi cell wall. So they're most effective as a protective, not a protective, but after your spore have been germinated in the leaf. And they're also classified as a medium risk to resistance development. And the last group that we are pointing out in this presentation is the MBC, which is a fungicide group that prevents cell division. And those ones, as the KOIs, are classified as a high risk to development resistance. So just to give you a couple examples on those, what products are available as for each of the groups that we've talked about. Also, it is important to talk that the Fungicide Resistance Action Committee have a classification for each of those with a code, and also they classify the risk of resistant development to each of those classes. And we can find out which group each of those fungicides represent in the label. For example, Quadris is a single mixture, a single active ingredient of azoxystrobin. So what that means is a group 11 fungicide. whatever. When we go through different products as Miravis Top, we have a mixture between two groups, and we have that pointed out in our label. So between those 4 groups, we also have mixtures between them to make our fungicide stronger. As we can see, we have mixtures between MCBs and DMIs. Also, we have fungicide mixtures between KOIs plus DMIs. Also, SDHIs plus KOIs, SDHIs plus DMIs, and also all-wave fungicide, which will be a mixture between SDHIs, KOIs, and DMIs.
Hello, my name is Ty Smith. I'm a PhD candidate with the University of Tennessee working in the plant pathology program. Today we will be discussing keeping fungicide resistance in check. After discussing what fungicide products are available, we will now discuss how resistance to these products occur and ways to delay that resistance. Generally, natural mutations are responsible for fungicide resistance to develop. This is not a function of the application of fungicides, but more natural mutations. These natural mutations can occur at a rate of 1 in 1 million chance in the field. Resistant isolates must already be in the field for these mutations to occur. The fungicide application is the selection pressure that selects for these resistant isolates. Applications select out resistant isolates while terminating sensitive isolates, causing the resistant isolate to be dominant in the field. To put this number into perspective, there could be roughly 18 mutant isolates of a certain fungi in one 40-acre soybean field. This diagram represents how we have selected for resistance with a fungicide group over time. Resistance occurs when populations that have a resistant mutation are selected for over time. As more applications of the fungicide are made, the resistant population slowly becomes the dominant population in the field. In this graphic, the red spots represent resistant isolates, whereas blue spots represent sensitive isolates. As the same fungicide is applied over and over, the sensitive population is slowly terminated while the resistant population remains. We'll now look at a case of fungicide resistance that is currently present in Tennessee soybeans using frogeye leaf spot. Frogeye leaf spot is caused by the pathogen Cercospora sogena and is only found in soybean. Frogeye leaf spot usually develops during reproductive growth stages. This can be blooming to maturity. But they may develop sooner in continuous soybean fields when under optimal environmental conditions. Frogeye leaf spot is characterized by angular or circular lesions with light gray centers and distinct purple to red-brown margins. Dark areas within the lesions on the underside of leaves are filled with clusters of spores known as sporulating lesions. The pathogen can overwinter on infested soybean debris and is readily spread by rain, wind, and on seed. It is prevalent in most soybean producing states in the U.S. Frogeye leaf spot was first reported to be resistant to QOI fungicides in Tennessee in 2010. A single mutation in the target site in the pathogen left QOI fungicides largely ineffective. There is also no visibly detectable difference between an FLS isolate that is sensitive and one that is resistant. The picture here shows the difference in colony growth between a QOI resistant and sensitive isolate in media with 4 different concentrations of the active ingredient azoxystrobin. The sensitive isolate, located in the bottom row, is controlled at 0.1 parts per million of azoxystrobin, while the resistant isolate located in the top row is only controlled at 100 parts per million. Since 2010, there have been 15 states, including Tennessee, that have reported QOI-resistant frogeye leaf spot in soybean. A mutation in the genome at the G143A position is the reason for this loss of efficacy to QOI fungicides. The photo shows the widespread detection of QOI resistance for Cercospora saginna or frogeye leaf spot populations. 15 states have detected resistance with the primary mutation being the G143A change. Resistance slowly spread up the Mississippi River Valley and is starting to progress outward to the east and west. The graph here illustrates the loss of efficacy of QOI fungicides over the years in Tennessee soybean production. 70% control was observed in 2013 for frogeye leaf spot infested soybean that was treated primarily with just a QOI fungicide. Since 2013, that control has decreased drastically to less than 10% if treated with just a single QOI product. The data shown here is compiled from multiple small plot fungicide efficacy tests from 2013 to 2019 conducted in Tennessee.
So talking about what we can do to avoid resistance or pretty much keep our fungicides working to control disease, we have pointed out some ideas and some actions that we can take to work with fungicide resistance. And one of them, it is enhance our residue decomposition with crop rotation, also do conservation tillage where it's possible. That's not actually possible on Tennessee farmland, but there are other farmlands where it's possible to be done. Also, there are general management practices as plant quality and disease-free seed. We can still do foliar fungicide sprays, but we have to go over different products, mix them with different active ingredients or with different fungicide classes. We can use resistant varieties, and the major one, which is very important to us, is scout for disease to use risk management strategies. We can see the majority of the practices that we can use to manage resistance, they happen before we even think about spraying a fungicide. They are majority practices that will be before we plant our fields and manage the spores that will work as a source of inoculum to infect our plants. And then the spores that later on will cause disease and will make us spray a fungicide, and then whatever we cannot control of those, they will remain in our fields as a source of inoculum and keep carrying on from the next season, spores that will infect our plants again, and it starts all over again. Therefore, our cultural practices and our resistant varieties are our major management strategies when we're talking about fungicide resistance management. And the fungicide spray is just a complement that we can do to those ones. And as we spoke, the fungicide resistance management, the use of a moderate or a resistant variety is one of the tools that is more effective to reduce FLS and also contribute to hold on the yield as a susceptible variety. Besides that, when we mix different fungicide classes, we can keep our population diversity, and then we won't be selecting only a part of the population that it is resistant, but we will keep our population as diversity as normal, so it will be much more manageable to work with disease control. And as we can see, when we do the adoption of moderate resistant variety and with a mix of fungicide classes, we can have great yield and low disease risk, as we can see in the graph, where where the bars and the line in gray are results for a susceptible variety. In green, we have results for a moderate one, and then on yellow, we have results for a resistant one. And our bars mean the yield that we've got, and while the lines means the disease severity for it. And for last, but the most important one, when we scout for disease risk management strategies, that's what we mean about do a fungicide spray only where it is required, and just if it is required. So as the University of Tennessee is searching for new ways to work on disease scout, and that by doing molecular work to know how much spores, what's the source of inoculum of your fungi that is present in the field and can cause infections, as well as work with thermal imagery to know if your plants are sick even before they show symptoms. And all of this data will be used to estimate disease risk and better target a fungicide spray. Therefore, as a summary for our presentation, we would like to state that KOI resistance to frogeye leaf spot pathogen is present in Tennessee soybean fields. DMI and MCB loss of sensitivity is under investigation, and the best what we can do to manage resistance is use cultural practices such as seed-free pathogens, crop rotation, enhance residue decomposition. Also, we can work on resistant varieties once that varieties range susceptibility from very susceptible to resistant. You can search by our QR code and go over the database for results on disease susceptibility to each variety that is tested every year in Tennessee. And also, we can use fungicide sprays and mixes between fungicide classes, and by accessing the QR code, you can have the information that's generated by the University of Tennessee for each product. You can also access more information on real-time disease and pest updates at news.utcrops.com. Also, you can use product guidelines on guide.utcrops.com, and also, again, go over the varieties database on search.utcrops and look over diagnostic tools, descriptions, and fungicides efficacy on guide.utcrops.com. Well, thank you very much.